Good day, friends, and welcome back to our channel. Today we are going to be discussing cultivation of viruses. So the question is, how are viruses cultivated? How do we culture viruses? Do we use culture media just as we use for bacteria? What are the ways? What are the methods? Is there just one method? These are the questions to be answered during the course of this video. So cultivation of viruses, how are viruses cultivated? Now, within the laboratory, viruses are cultivated using three major ways. Three major ways are used to cultivate viruses. Now, the first way is what we call animal inoculation, by inoculating the virus into animals. The second way is by inoculating the virus into embryonated eggs, eggs of chickens that have embryos. The viruses can be inoculated into such medium, such eggs. And the third method of cultivating viruses is by inoculating the viruses into cells or tissue cultures. Now, these are all important ways, but like I said earlier, the viruses are obligate intracellular parasites, and viruses, a fully matured virus particles is what we refer to as a viron. Now, the question to also ask is, what is the purpose for cultivating viruses? Why are we cultivating viruses? Are we trying to cause more disease? No. The reasons for cultivating viruses is, one, to be able to identify a virus that is present in a clinical sample. If somebody brings a sample, sputum, blood, feces, and brings it to the hospital or to the laboratory, the scientist wants to be able to identify what kind of virus is present in that clinical sample. So that is one reason for cultivating virus. A second reason for cultivating virus is for research purposes. We want to be able to understand virus, understand viral structure, viral nature, viral function. All of this can only be done by cultivating the virus, and this is very, very important to note. Now, it's also important to note that viruses can be cultured or cultivated for the reason of producing vaccines. A lot of viral vaccines have been made. There's vaccines for COVID-19 and so many vaccines like that. So many at times, these viruses need to be cultivated. They need to be cultured to give us, materials that can be used for the production of vaccines. So these are three major reasons why we culture or cultivate vaccines. Now, going to the actual cultivation of vaccines, we are going to start in this video just discussing animal inoculation. That is one of the ways. We'll discuss the other two ways in subsequent videos. Now, animal inoculation. What is animal inoculation? Animal inoculation means that for the virus to be cultivated, this virus will be inoculated. Inoculation is a fancy word, a science word for injecting or inputting this virus into an animal. Now, there are many kinds of animals that viruses can be inoculated into. Viruses can be inoculated into pigs, into rats, into mice, into all sorts of primates, and this is very, very important to note. Now, viruses are usually inoculated into mice, especially mice which are just about 48 hours old, and these mice must be free of illnesses, not mice that are diseased. Usually that is the way, or these are the animals that are used for the inoculation of viruses, so that we can cultivate viruses in these animals. Now, these viruses are usually inoculated in quite a number of ways. Number one is intranasal. Intranasal meaning by allowing the animal, the mice or the organism, to inhale the virus, so through the nasal or the nostrils. So that kind of inoculation is called intranasal. Now, we also have intracerebral. In this kind of method, the virus is injected into the brain of the animal. So that is the intracerebral route. We have the subcutaneous route, which is the injection of the virus or the inoculation of virus through the skin of the animal. And then the fourth note is the intraperitoneal route. The intraperitoneal route. Every animal has a cavity, a hole within the body, which we call the body cavity or the peritoneum. So the injection of the virus into that cavity, into that peritoneum, is what we call intraperitoneal inoculation. So these are some of the ways through which the viruses are inoculated or injected into these animals for their growth and their cultivation. Now, what are some of the advantages of using animal inoculation as a method of cultivating viruses? Some of the advantages is that it's easy to use, it's easy to help, it's easy to use, it's an easy way that helps scientists to be able to diagnose different viral infections. It's an easy way for scientists to use to study pathogenesis. That's the way a disease develops. Now, antibodies are also formed very easily by the body against viral particles when animal inoculation is done. So it's easy to take samples from the mice and get antibodies for that viruses. And it's an easy method that can be used to study oncogenesis, that is the beginning or the cancer genesis. So these are advantages for the use of animal inoculation as a method of cultivating viruses. There are disadvantages of using animal inoculation is that it's expensive and it's also difficult to maintain the animals. You have to feed them, you have to care for them. So these are some of the disadvantages of using animal inoculation. Thank you for listening to this video. I hope we have an understanding of what viral cultivation looks like. In subsequent videos, we'll talk about other methods. Please like our content, share our content, and watch many more of our videos. Have a wonderful day.